All right, on to the 3 steps of cellular respiration. So the 3 steps are outlined here in this diagram, and this is a really nice diagram because it shows you not only the 3 steps, but it also shows you where they occur. So we're going to start with glycolysis, and that occurs in the cytoplasm, And that is when you are taking glucose and you are converting it through a series of reactions, series of steps, hence the 3 arrows. There are more than 3 steps, but you get the picture, into something called pyruvate. Okay, so you're actually going to cut the glucose into 2 molecules of pyruvate. And again, this is occurring in the cytoplasm. And then the pyruvate goes into the mitochondria matrix in particular, and that is where something called the Krebs cycle occurs, also called the citric acid cycle. We'll use both just so that we cover our bases in terms of this cycle. And in this cycle, we use the pyruvate to make lots and lots of NADH and another molecule, which is an electron carrier called FADH2. All right. Oh, now back here in glycolysis, we do make some NADH as well. So NADH, FADH2, all of these, kind of building them up in these first 2 steps to be used in the final step, electron transport chain, oxidative phosphorylation. And this is where we are going to use the energy in those electrons to power the production of ATP. All right, and that too occurs in the mitochondria, this last step. So let's go on to the first step. Now all of the reactions are shown here. So here we start up here with glucose, and it looks very complex, I can understand, if you're a little overwhelmed, but what I really want you to get from these diagrams is we start with glucose, and glucose is converted by an enzyme. Oh, there's an ACE, thank you. And but converted by an enzyme to an intermediate. And we need to put a little bit of ATP in to do this, to convert glucose into, what is this, glucose 6-phosphate. And then another enzyme converts it into another intermediate, and then another enzyme converts it into another intermediate. Oh, we need a little bit of ATP there in order to do that conversion. And then another enzyme converts it to an intermediate that is actually an isomer, or the isomerase, kind of back and forth between these 2 forms. And then we'll head up this way. And the conversion of the glyceraldehyde 3-phosphate to this other intermediate, what is it, 1,3-bis-phosphoglycerate. In doing that, we put some electrons onto NAD+ to make some NADH. Yay. And we actually make 2 of them here. And then we go from that intermediate to the next intermediate. We actually produce some ATP in this process. Okay, 2 ATP. And then we go from one intermediate to another, and then the final step here, we produce a little bit more ATP, and we finally get our 2 molecules of pyruvate. So the only molecules I really need you to know from these diagrams is, of course, glucose. We're starting with glucose, and we're ending up with pyruvate. And so all of the intermediates here, you would have to know those for, let's say, a biochemistry class, but we are not going to concern ourselves with the in particulars of the intermediates or the particular enzymes that convert one intermediate to the next. But what I would like you to appreciate and understand is that we have a series of steps over here, this first few series of reactions that require energy. So we're investing energy into these first few reactions. That's the energy investment phase of glycolysis. And then over here, in the second series of reactions, we're actually making some ATP. That's a payoff. So we put in 2 over here. So we're in debt by 2 ATP molecules. But over here, in the second half of the process, we're making 4 ATP. So our net ATP considering the ones we've used and the ones we've made, we've actually net 2 ATP for this process, which really isn't very much and really isn't the point of glycolysis. The point of glycolysis is to make pyruvate. That is the major product. Okay. And we do make a little bit of NADH. Thank you. Okay, that's great. And we will use it, but it's only a tiny bit. And again, it's really not the major focus of glycolysis. The major focus is our pyruvate. We've made 2. So we've gone from a 6-carbon sugar to 2 3-carbon sugars. We've split that sugar in half. Okay, so the reason the pyruvate is so important is because we need it to start the Krebs cycle. If we don't have pyruvate, we can't start the Krebs cycle, also called the citric acid cycle. Okay. So with the citric acid cycle, we're going to take the pyruvate